You're watching CTOM TV. Knowledge is power. You'd like to tell us about CAR T cell therapy. Now, this is something I know absolutely nothing about, so I'm interested to learn along with everybody else here today. So how would you like to proceed?
It's been a lot of work. I haven't spent a lot of time on CAR T. I did do a lot of immunology in my undergrad years. In fact, I almost did a complete degree in it.
Didn't a complete degree in which?
In immunology.
Okay.
I did my fourth year in advanced immunology courses.
All right.
So it was a big, definitely a big interest of mine, and I have spent a lot of time on it. We spend a lot of time on various types of immunotherapy, but the CAR T is a whole new area of immunotherapy that I haven't spent a lot of time on over the last few years.
Sure.
It involves a T cell receptor.
Right.
So I have to go back and relearn a lot of things.
And you're going to tell us why. I mean, we had a brief chat in anticipation of our show today, but there are some reasons why it's not, being offered. So it's not so accessible just yet.
It's very expensive.
That's right.
And sorry, don't care. There are some significant hurdles. It's not the easiest thing to do, and does have some side effects.
Okay. All right. So we want to hear about all those things, but
yes.
But we'll start with what it is, I guess.
Yeah. Well, I think we need a bit of an immune system primer before we get started.
Okay.
So what I would like to do is I'd like to talk about the immune response in regards to cancer.
Okay.
Briefly. And then talk about what CAR T cells are.
Okay.
Why they're different from our regular immune cells. And then I would like to talk about a couple of drugs that are involved, and I've taken some notes on those, some of the newer drugs. I've got a couple of diagrams I'd like to share. And then we're going to talk about some of the problems, some of the hurdles, and then some of the solutions to that.
Okay.
So to start out with, Immunotherapy 101. How does the immune system interact with cancer cells?
Okay.
So cancer cells, when they die, they release things called antigens. An antigen is referred to in the most basic term as an antibody generator. That's what the term antigen stands for.
Okay.
Antibody generator. And so anything that causes our bodies to produce antibodies against it is an antigen. And typically these things are proteins on the cell surface of cells that are foreign to us, that our body does not recognize or is growing incorrectly. So cells have these antigens. Now, antigens can be on the surface of the cell where they can be recognized, or they can be on the interior of the cell, like a mutated genetic pathway or something, or an altered protein. So what happens is when cancer cells die, they release these antigens. I mean, there's many processes where they release these antigens into the blood, including direct contact with the immune system. But these antigens are taken up and presented on the cell surface of cells called antigen-presenting cells. Okay. They're referred to as APCs. And basically what these do is they say, hey, this is a foreign invader. They're kind of like the call center, the 911 call center. They say, hey, they take this call. Hey, here's this foreign invader. This is what it looks out. We're going to put a bulletin out on the surface of all of our cells, and we're going to send this off to the army or the police force. And the police force is basically what we call T cells and B cells as well, but mostly T cells. And these T cells will take this antigen. They'll then go out and seek out any cell that has that antigen and attack it in a variety of ways. So basically what happens is these APC cells, these antigen-presenting cells that have a piece of this tumor antigen, they'll go into the lymph nodes and they'll activate the T cells and teach them to recognize these tumor cells. And they do this through something called, number one, a T cell receptor, which is a bridge between a T cell and any other cell. It's an external receptor. and also this co-stimulatory pathway called a CD3. And this CD3 is basically attached to the T cell receptor. So the T cell receptor will take the antigen from the antigen presenting cell, which is presented in a format called MHC1. We don't need to worry about that. It's called major histocompatibility 1. It's just a way of, it's like a receptor that presents this antigen to the T cell receptor.
Okay.
And it requires that presentation. So the T cell is not just going to take an antigen out of the blood or off a tumor cell. The T cell requires it to be presented in a edited way via this MHC1
Okay.
from an antibody presenting cell or something. So then the T cell gets this peptide or whatever it may be,
The antigen.
Yeah, the antigen. And it gets it through the T cell receptor. And then at the same time, these antigen presenting cells, this all happens in the lymph nodes, the antigen presenting cells turn on the T cells as well. They say, here's this, now here's a switch to turn you on, so you make many copies of yourself. So then the T cells divide and grow. And you have a big mass amount of them, and then they go out and seek out that antigen.
Okay.
So then the T cells travel. They go through the blood vessels to reach the tumor. They infiltrate it, recognize the cancer cells, and kill them.
Okay.
So that's the standard way our immune system interacts. So the types of cells that we have, now they all have CD3, which is the switch that turns it on. We have cytotoxic T cells, and they directly attack the cancer cells. We have helper T cells. So the cytotoxic T cells are typically what we call CD8 positive cells.
Mm-hmm.
The helper cells, they recruit the immune system and they organize a response. Then we also have regulatory T cells, and they suppress the immune system so that it doesn't overact, like in autoimmune disease.
Mm-hmm.
So in other words, you don't want it to just kill everything in its way. So they provide some suppression. So these are called Tregs, and they are usually CD4 positive. So the main players in cancer are the cytotoxic T cells that are labeled with a CD8, and then the regulatory T cells that have a CD4. And then we also have memory T cells, and they remember the antigens of cancer cells for your lifetime, basically.
Okay. So the helper cells, are those the Tregs? Is that a separate thing?
Helper cells are separate from the
Okay.
Tregs.
Okay.
It's a very coordinated system that requires many players.
Sounds like.
Yeah. So then we have CAR T cells. So as you can imagine, a T cell with a T cell receptor is not always going to work with cancer, because cancer's been able to override certain features of it. Cancers are smart. They've been interacting with our immune system for many, many years, and they're able to evade them and turn them off and use, they're able to, for example, they're able to use regulatory T cells, Tregs, and use the growth factors and have them feed them instead of the actual immune system.
Mm.
Yeah, so I'm going to get you to cue that first slide, and I'm going to show you the difference between T cells and CAR cells, CAR T cells.
Okay, so let me see here. Just a sec, folks. I want to share here this slide share. All right.
So here we've got a diagram of a T cell receptor on a normal T cell and a CAR. Now, what is a CAR T cell? CAR stands for what we call chimeric antigen receptor. And it's a T cell. So it's a T cell that now has what we call a chimeric antigen receptor. And so what does that mean? This means that it is engineered to have the ability to target a specific protein. So we're taking away the function of this T cell receptor from it being a inquisitive thing that can go around and function in the immune system. We're saying, hey, we're taking over your function. We want you to do one specific job. We don't want you to do the many jobs you already do and protect the immune system. We want you to just destroy anything that has this tumor antigen.
Okay.
So what we do is we combine both antigen binding T cell receptors and T cell activating functions into a single receptor. So not only we are able to now turn it on, so now what we have is we've changed the receptor so it binds a certain particular antigen, and it doesn't have to be presented by the MHC1 now. It can just be on the surface of the tumor cell. And when it does bind that, it doesn't need to be turned on by these antigen presenting cells. So we're basically bypassing the whole function of the immune system.
Right.
We're bypassing the antigen presenting cells. We're bypassing the helper T cells. We're bypassing all of these other cells and going straight to the chase. And so basically what we're doing is we're taking tumor cells or T cells out of the body. We're teaching them to, or we're creating a gene, a receptor in them that will target any cell that has a specific antigen that patient has in their tumors. So as you can see, these T cells have a variety of different, the T cell receptor has a variety of different functions there that allow it to function in a proper manner. When you go to the CAR T cell on the right, you see it's much simpler receptor. It's taken a lot of receptor from the antigen presenting cells or the B cells as well. And so it's got this multiple function where it's able to just bind onto, as you can see, it binds the tumor antigen. Here, the antigen has to be presented by the MHC1. In a tumor, we don't need to do this anymore with the CAR T cell. So it's direct targeting.
Right.
So how do these cells different? How does a CAR T cell? Now I should say, CAR T cells also have a functional normal T cell receptor. So they have both. They have the CAR T cell receptor and the normal receptor.
Okay.
So they're not just they're chimeric. They have both in that sense too. So T cells versus CAR T cells. So antigen recognition. So the T cell receptor in normal cells will only recognize an antigen if it's presented by the MHC1, as you can see there. Whereas a CAR T will find anything that's expressed on the cell surface that is designed to target. Doesn't need to be presented by MHC1. As far as restriction goes, the T cell receptor will take the antigen, which is the same between different people, and the MHC, which varies between people. So it won't go after things that it's not sure about or that are self. The CAR T only goes after the antigen. So it's able to perform off-shelf targeting of antigens. So the big difference between these two is T cell receptors in a normal functioning T cell can recognize thousands of intracellular targets. That means targets inside of the cell, because the process that the immune system uses to identify an antigen can often be from something inside of the cell. So it will take a piece of DNA from the tumor or a piece of DNA from the virus or bacteria or something from inside of the cell. So it can use thousands of these different things from inside of the cell and display them on this MHC and teach the immune system to go after those cells. So it's a very adaptable, functional process. And then also, because a T cell receptor has this MHC1 presentation, there are lower risks of side effects. It's not going to have any off targeting. And the other thing about a T cell receptor is because of those memory cells, these T cell receptors that will target a certain protein or virus or cancer or something typically last forever. They're always there in your body. So, when you get that vaccine, it lasts a lifetime. Now, a CAR T cell receptor is a little different. So they only recognize about 10 different surface antigens. Because of this internal processing system of the MHC1 and the intracellular targets, CAR Ts bypass all of that. They can only recognize things on the surface of the cell. And so because they're MHC independent, they can't tell the difference between a healthy cell or a tumor cell. They only go after what they're designed, what that T cell receptor, that chimeric T cell receptor that you've genetically programmed will go after. And also the host can produce anti-CAR T antibodies. So your body could see these CAR Ts and say, hey, these T cells are broken, let's get rid of them. And it will actually create antibodies to attack the CAR T cells because they have this altered receptor.
Yes, they seem like foreign.
Yeah, so you can also get something called cytokine release syndrome, which is a dangerous side effect. And then CAR T cells typically only last about 10 years in the body, maximum.
Okay.
Yeah, so let's go to that next slide and we'll talk about how CAR T cells really work. So what happens, as you can see in this slide, is you'll start with, they'll take your blood and they'll isolate the T cells. Then they will isolate these T cells, and what they'll do is they will take the gene that creates this chimeric antigen receptor, this CAR T receptor. And so they have a gene for that. They've created a gene in the lab for that. They insert it into a virus, and this is just standard cloning. I mean, we've all done this in the lab many, many times. You can insert genes into a genome quite easily. It's not a difficult process. And so what they do is they take these isolated T cells from your body and they insert the gene. So this is genetic engineering. They insert the gene for this altered T cell receptor. And so they can insert a gene that will target any kind of antigen, literally. Right now, the antigen that they target is something that has to be unique to cancer cells and not in normal cells. And so the best target they've used so far is something called CD19. It's present on the vast majority of leukemia B cells and so on, and lymphoma B cells and so on. And it's also present on some healthy cells, but not all of them. And so it's the best target that they found so far. They're also looking at different targets, but the cell surface antigen that they use in most CAR Ts is CD19.
Okay.
So they basically insert this CD19 targeting CAR T receptor into your T cells through a viral vector, standard cloning, and then they'll grow it up. And you have these modified CAR T cells, so grow them up in a petri dish, and then they'll inject them back into you. And so as you can see in step 4 there, they will find the cancer cell that has that antigen and they will bind onto it and break it up and attack it. Yeah, just simple as that.
Wow.
Yeah, so it's a very simple process when you look at the lab techniques and so on involved. I mean, cloning is something that we've all done. It's quite easy to do.
Well, I have not, but I understand.
Well, if you've taken a second year biology course, you've done cloning.
Okay, all right.
Yes.
So the process, so it was, it's really kind of finding what you're looking for, and you're saying for the most part, they are looking for the CD19.
Yeah, and so the biggest limitation is finding something that is on the tumor itself.
Right.
So, the biggest problem is most tumor antigens are internal, and CAR T cells could not recognize something that's internal because they skip this whole antigen presenting cell technique process and this MHC1 process.
Yes, that's right. So you were saying the regular T cells can go inside the cells and they could find thousands of different.
No, they don't. They're just the attackers. They're the police force.
Okay.
It's the other cells that go in and get that information, the antigen presenting cells.
Okay.
So first, actually, it's a more complicated process. You have these things called dendritic cells, and there's different cells, macrophages and so on, that will break up the cancer cells. Or if the cancer cell antigen is floating around in the blood by itself, it can get taken up by APC cells. So that's a 3-step process. Okay. First, you have the damaged cancer cell, that has been attacked by the immune system. Then you have the release of the antigen presenting cells find the antigen, process it through a process called TAPS, which is a process called transportation of antigen presentation. That is a process where the antigen presenting cells will process it and then put it on the MHC1 receptor, and then they will give it to the T cells that are the police force that will go out and attack it.
Okay. And in contrast to that process where thousands of things inside the cell can be detected,
yes,
the CAR T can, as you say, only recognize about 10 different things,
yeah,
and that's on the
so
outside, that's on the
cell,
surface.
exactly.
So the CAR T is kind of like zombie cells, right?
They are,
they've been kind of, the brain's been taken out of them.
Exactly.
They have one purpose,
Exactly.
and they're not free.
CD19.
They're not free thinking.
No, they're not.
I see.
Yeah.
Okay,
and that's the problem with them.
Okay.
They're not intelligent like the immune system,
sure,
or adaptive.
But they do the job.
They do the job, yes.
Yeah.
They're like drones.
Yeah.
You know, very much like drones.
Oh, that's interesting.
Yeah. So I'm just going to briefly talk about some of the FDA approved treatments right now, CAR T cell treatments.
Okay.
So one of the popular ones is Kymriah. It's FDA approved for patients under 25 with refractory B-cell precursor acute lymphoblastic leukemia, ALL.
Okay. So this is an example.
Yes.
Okay.
This is a drug that's FDA approved.
Okay.
In these patients, it gets a wonderful response rate. I'm not going to talk about the response rates because it's a variety of factors here, but I'm just going to tell you briefly what they are.
Okay.
And so it's approved for ALL. It's also recently approved for adult relapsed refractory diffuse large B-cell lymphoma. And this is really great because we haven't really had anything for large cell lymphoma. And what large cell lymphomas are, or diffuse large cell lymphomas, DLBCL, they're typically what happens to a B-cell lymphoma when it's untreatable. It develops these diffuse large B cells, and when that happens, there's really no treatments for it. So having this as a treatment, now there are some other approaches that we can talk about at some point in time, but as far as CAR T cells, they've actually been FDA approved for this condition.
Okay.
So this is one of the first real treatments we've had for patients that have advanced lymphomas that haven't responded to therapy and that have progressed onto large B-cell lymphomas.
Okay.
And so it's also FDA approved for high-grade B-cell lymphoma, DLBCL, that has progressed from something called follicular lymphoma.
Okay.
So when you get DLBCL, there's two subtypes. There's the germinal center, and then there's the ABC activated B cell. There are two subtypes, and one has a worse prognosis than the other. And they typically come from different types of lymphoma. So in this particular case, the CAR T cells work quite well on the bad form of it, which is derived from follicular lymphoma. So there's also another one that's been FDA approved called Yescarta with a Y. And so that's been approved for adult relapsed and refractory diffuse large B cell, DLBCL, and also the same DLBCL that's been derived from follicular lymphoma, the bad type. Now, one of the side effects with CAR T is what we call cytokine release syndrome. And it's been a real problem, and it's killed some of the patients. It's a bad side effect. It prevents patients from being treated, and it's also something that kills people. We'll get into what that is. But recently there was a drug approved for that called Actemra. And it's actually approved for anyone on CAR T cell therapy that has life-threatening cytokine release syndrome. So this is a solution to a dangerous problem.
Oh, that's good.
Yeah.
It takes one of the downsides.
One of the downsides,
right?
yes, very significant.
Like the other downside you mentioned is that it only lasts about 10 years, but you know,
the memory of it, yes, exactly.
Yeah. But if you can get around that cytokine release syndrome, that sounds positive.
Exactly. Yes. So one of the big things is the costs. You know, and this has really been something that has prevented people from getting treated with CAR T.
So let's talk about that. What is the standard cost?
Standard cost in USA right now is about 440,000, 100,000. So, you know, almost
$440,000.
Yeah, so almost
Holy
almost
schmoly.
$500,000.
Wow. So it's not a treatment everybody could afford, to say the least.
No, yeah.
Okay.
Now I see this going down significantly, and this technology is something that, you know, sure, it's got to be done a certain way, but these are standard techniques.
As you say, second year biology,
we do cloning. Yeah, it's not quite that simple, but the techniques are the same. Okay. You know, it's like if you can drive a car, you can drive a truck eventually with more training, or an airplane with even more training. So, yeah, the cost basically is, you know, just under $500,000, or has been. That is going down. Now, I did a bit of research. I thought, okay, well, since this is something that a lot of hospitals and labs are probably working on themselves in-house, is this treatment available elsewhere? And interestingly, it is. China is doing a lot of it, and so is Israel. And I looked at some of the private clinics doing it, and they had costs. You know, they're basically copies of these other two drugs. They may even be using these drugs, but I suspect they're doing it themselves. So some of the costs are down to between 90,000 to 110, 120,000 US in China and Israel. And Israel has a phenomenal medical system. China, you know, I think is really coming along. I don't know how well they're doing. I think, you know, we'd have to look into that deeper before we'd start recommending it. But I looked at a couple of treatments. So in China and Israel, they have a B-cell lymphoma treatment, CD19 CAR T therapy, and they're claiming they're getting a 90% complete remission rate.
Wow.
Yeah, which is fairly standard for CAR T therapies.
Wow, that's the one thing that we haven't talked about yet. But that 90%.
Well, that's what these drugs are getting. They're really blowing things out of the park.
That's why people might pay $440K if they had it, because holy cow, 90% full remission.
And then there's another one for B-ALL, B-cell acute lymphoblastic leukemia, and that's in Israel and China. And they're getting, it's a CD19 CAR T, and they're also getting a 90% complete response rate.
So that's a pretty good risk then, you know, if you could, you know, $90,000 to $110,000. And I appreciate that's still a very select population of the planet who could do that for sure.
Yes. Now, there was another clinic in China that has developed one for multiple myeloma. And they're using something called a BCMA CAR T. And I don't know, you know, what the statistics were for this, but they claim to have 82% response rate with long-term survival. This is just off their website. I didn't quite, you know, go into the details and look at their peer-reviewed data, and that would also always be required. But, you know, I think the point is that people are doing this, and we're going to see a lot more of it.
Right, and the prices are coming down.
And now also in Israel, they're offering CAR T CD19 for acute myeloid leukemia, AML.
Wow.
So that's really something.
Okay, so B-cell lymphoma, B-ALL, MML, and AML.
Yes, AML. It's not MML, it's MM, multiple myeloma.
Multiple myeloma, MM.
Those are your initials, aren't they?
They are. Wow, that's great. 82% is the worst statistic remission for these people who would otherwise be out of options.
Yeah.
Okay. So that answers the question why this has continued to proliferate as an option, even with that outrageous price, is because holy cow, look at what it's providing for people.
Yeah. Now it's specific to you know, these lymphomas and leukemias. They haven't had much success with solid tumors, and the reason is that a lot of the antigens for solid tumors are internal.
Okay.
And on top of that, it's hard to get an immune cell inside a dense tumor. Okay. You know, solid tumors are hard to get into. So it's hard to get those cells in there.
Okay, but for the leukemias and lymphomas, here's an option.
Yeah, totally.
Wow.
So I'm going to talk a bit about problems and side effects.
Okay.
Okay, so relapse is the biggest problem, as you can imagine. And so the causes of relapse in CAR T therapy is sometimes the tumor cells lose expression of their CD19 cell surface receptor. And if that happens, the CAR T cells are just going to ignore it.
Yeah.
They're not intelligent enough like a activated T cell. So they're just going to ignore it. Brainless drones, as you said.
Yeah, so they just kind of walk right by it.
Yeah. And then there's limited persistence of CAR T cells, where the CAR T cells just don't actually flourish in the body for whatever reason. It could be due to the immune system creating these anti-CAR T antibodies, and recognizing it as foreign. So typically what happens though when you get CAR T cell therapy is you get a suppressive chemotherapy to suppress your immune system. Obviously, that's required when you inject a new type of T cell in your body.
Okay.
So, they have to kill off the immune system to a certain degree before they give this. So it's almost like a stem cell transplant, but not quite. So, sometimes that chemotherapy doesn't work as well and doesn't kill off all of your immune system, and your immune system starts killing your CAR T cells.
Sure.
And then your immune system and your body can just inhibit the activity of CAR T cells. And then, once again, one of the biggest problems is that most tumor antigens are internal. So some of the side effects of CAR T. So cytokine release syndrome, we talked about that. And we've talked about that drug that's recently FDA approved for that. So that's, it's not such a big problem.
Actema.
Yes. So one of the issues with cytokine release syndrome, it's kind of important because it shows that the CAR T is working.
Okay.
The higher your CRS is, your cytokine release syndrome, basically if you have a high disease burden, that means a lot of cancer, then you're going to get more CRS. So it's based on your disease burden.
Okay.
Obviously, too much can be deadly. And we talked about one other drug, tocilizumab. We didn't talk about that drug, but that's what's being used currently. And I mentioned the other drug. Let me get back to it. What is it called?
Yescarta?
Yes. No,
Actema.
Actema, which is tocilizumab.
Okay.
Tocilizumab.
Say that fast 5 times.
Tocilizumab.
Just kidding. But good effort.
I can't keep up with these drug names. I don't know where they get them from. And they name them different names too. They're not all the same name. For the drug can have multiple names, same drug.
Depending on the country and whether or not, yes.
So another problem is what we call macrophage activation syndrome. This is often associated with cytokine release syndrome. And it's the excessive activation and multiplication of T cells and macrophages. We can also have tumor lysis syndrome, and we've seen this a bit in PD-1 inhibitors. That's where you get a massive amount of tumor cells dying and releasing their contents into the body, and that can be toxic and poisoning. Yeah, it can really cause bad side effects and so on.
Sure. Well, you're messing with a lot of machinery in there when you're doing this genetic altering.
So we can also get anaphylaxis, which is a life-threatening allergic reaction that's just often associated with peanuts, but this can happen as an immune response against the CAR T cells. We can get on-target, off-target toxicity, and that's because many other tumor antigens are also expressed in healthy cells. We can get B cell aplasia, which is low or absent B cells, and B cells are an important part of the immune system. And so the CAR T cells can destroy cancerous and normal B cells. Now, obviously, if you have a B cell lymphoma that you're targeting with CAR T cells, you want to see those cancerous B cells dead, but you don't necessarily want to see the normal B cells dead,
Mm-hmm.
because then that's destroying your immune system.
Mm-hmm.
So it is an indicator of CAR T cell activity. And then finally, there have been cases of neurological toxicities with CAR T cells.
Mm-hmm.
So, you know, there are a few issues, definitely.
Mm-hmm. And yet, because of these outrageous
Response rates.
not 90, 82%,
Mm-hmm.
full remission.
Yeah, it's incredible.
I can appreciate why, if that's my only option and I had the money, you know, 440K in the US, maybe 90 to 110 in China and Israel.
Go to Israel, yes.
Go to China.
Yeah.
I can understand why people would be
I would definitely
perhaps
trust
willing.
CAR T in Israel. I mean, you know, Israel has some brilliant scientists.
Mm.
They've been leading science and technology for a long time.
Mm. Well, there you go. So there's all these side effects, but again, if that's your only option, and with those kinds of statistics of what the outcome could be, I could appreciate why someone would seriously consider it.
Yes,
Mm-hmm.
I would.
Yeah. Yeah.
So that's it. That wraps up our
okay.
CAR T.
Wow. Okay. Well, it makes sense to me, though, you know, at that price and with the process, why it's not, you know, mass produced just yet in the way some targeted therapies are. And exactly.
And we're going to see many more different antigens targeting.
I'm sure. Yes, that makes sense in time. I mean, I'm just thinking in the last 10 years, what we've seen in terms of the massive increase in numbers of targeted therapies and the great reduction in price for genetic testing and for these treatments. It's definitely coming. Yeah. Okay, great. Thanks, Alex. Thank you for doing all that research so you could share all that information with us about the CAR T therapy, for sure. Now, next week, folks, Alex is going to tell us all about the latest and greatest treatment options for cervical cancer.
No, we're just dealing with cervical cancer because there's a few other cancers directly associated with cervical.
Well, that would be entirely up to you to determine.
Okay.
What I have in front of me is next week cervical cancer. So why don't you just show up and present whatever you'd like us to know, whatever you feel is most important in relation to that. And if we need more time, we can always add more time on the following week or something. I know sometimes these things have like 3 or 4 layers or different.
Well, cervical cancer is fairly restricted to HPV infection. Yeah.
Okay. And so, okay, well, you can bring whatever you want. Ovarian, just let me know ahead uterine, of time.
Fallopian.
Okay. Sure.
We'll look at that. Why don't we look at all of those?
Why don't we? Because,
with an emphasis on cervical.
Sure. Okay. So say that again. We've got this fallopian, you said?
Yeah.
Cervical.
Uterine.
Okay. Okay, great. Well, you have just given yourself more work, my friend.
Female reproductive cancers.
Okay. That's what we're going to call it. Okay. This is why you're the cancer guy, because you know all this stuff. Female reproductive cancers.
I don't have those organs.
No, you don't need to have the organs, I suspect, in order to understand
Probably not.
The process of the cancer within it and how best to treat it. Okay, so next week, female reproductive cancer. You heard it here first, people. So we'll be here next Thursday at 1 o'clock Pacific Standard Time. We'll answer any of the questions that have come through the week. Anything that you share with us live, we'll make sure that we address, and then we'll cover that topic. Between now and then, if you want to find out more information about what's possible in cancer care specific to a type of cancer you or someone you care about has, do please visit Alex's YouTube channel, thecancerguy.com. You'll find literally hundreds of videos on all different types of cancers and key elements of precision oncology, otherwise known as personalized cancer care. Different types of genetic testing and other diagnostic tools, targeted therapies, immunotherapies, even some good science about lifestyle changes in diet and exercise. It's all there at thecancerguy.com. And if you or someone you know has cancer and you'd like to chat with Alex and his team and find out what can be done for you, you can find out how to reach him at ctome.com. That's Cancer Treatment Options and Management. ctoam.com, and you can just reach out and let us know that you'd like to chat, and we'll be happy to speak with you. So we'll see you next week. Have a good week.
Bye.
Thank you for watching CTOAM TV. Subscribe below and stay informed.